So next we're going to talk about the functional units of the kidneys. So what does when we think about the kidneys and the main function of maintaining, regulating the composition, pH, and volume of body fluids, what does that? So the functional units of the kidney are called nephrons, and there's about 1,000,000 nephrons per kidney. So the nephron is going to be made up of a renal corpuscle and a renal tubule. So if we look over here, the structures, the granular structures up in the cortex, those are all renal corpuscles. And then we have the renal tubule that kind of, depending on if we're talking about cortical nephrons or juxtamedullary nephrons, the bulk of it is going to be located in the cortex with just a portion of the loop of Henle dipping down into the renal pyramids. So over here, this is our renal pyramid. This is the cortex right above it. These guys right here, those are the renal corpuscles, okay? And then all of this highly coiled tubule, that's the renal tubule. So that's where we're going to identify the structures of the renal corpuscle and the renal tubule. All right, so first off, let's do the renal corpuscle. So over here, this is an enlarged model of the renal corpuscle. Let's talk about the structures of the renal corpuscle. So first, in the last video, I mentioned this is the glomerulus. So it's a specialized tuft of capillaries, and these capillaries are special because they're fenestrated capillaries. Fenestrae actually means quote-unquote window, like little window. So these capillaries have holes or little windows that allow for substances to be filtered out. So it makes this ideal for a filtering capillary, okay? So this is the glomerulus, a specialized tuft of capillaries. Now, directly, the renal corpuscle itself is going to be made up of a visceral layer and a parietal layer, okay? So with the visceral layer, the visceral layer is closely associated with the glomerulus, and it's made up of these highly specialized epithelial cells called podocytes. So these podocytes line the glomerulus, and they have these primary processes that extend from the cell body, and then all these little secondary processes that extend from that. And those secondary processes are called pedicels, and a pedicel literally means like little feet. So what happens are these podocytes have these little pedicels that interdigitate with other podocytes and then create what's called slit pores or filtration slits. So together with the fenestrated capillaries of the glomerulus and the visceral layer of the renal corpuscle, the podocytes, together we have what's like a filtration membrane, okay? So it creates a size exclusion, right? So things that are small enough to get through first the fenestrae of the capillaries and then through the slit pores or the filtration slits of the podocytes, that is what's going to accumulate as the filtrate in this renal corpuscle, okay? Anything that's larger than those fenestrae or those slit pores will remain in the blood, okay? They won't be filtered out. So that's how we get our size exclusion. So things like large proteins, plasma proteins, those should never end up as a part of the filtrate. Those are going to be too large to fit through the fenestrae or the slit pores. All right, so that's the visceral layer of the renal corpuscle. The parietal layer is made up of a squamous epithelium, and it's this layer right around here. This is called the Bowman's capsule, okay? So the Bowman's capsule or the glomerular capsule, that is the parietal layer of the renal corpuscle, okay? This space between the glomerulus and the Bowman's capsule, that is where the filtrate will accumulate. So everything that gets pushed out of the blood because of that high filtration pressure is going to accumulate in this Bowman's capsule and then eventually make its way through this first part of the renal tubule called the proximal convoluted tubule. It's proximal because it comes directly off of the renal corpuscle, convoluted because if we look over here, here's our renal corpuscle. This is the proximal convoluted tubule, and it's highly convoluted, okay, highly coiled. So this is the proximal convoluted tubule. When it straightens out and starts to go down and dip a little bit into the renal pyramid, this is called the loop of Henle. We have our descending loop of Henle, then our ascending loop of Henle. Okay? When it starts to be coiled again, up here, this is what is called the distal convoluted tubule. Distal because it's further away from it's not directly connected coming off of the renal corpuscle itself, but it's more close to the collecting duct over here, okay? So again, here's our renal corpuscle. This would be our proximal convoluted tubule. This is our descending loop of Henle because it's dipping down. Then it curves back up and is now our ascending loop of Henle. Here's our distal convoluted tubule, and then here's our collecting duct. Okay. Now, the renal corpuscle, proximal convoluted tubule, loop of Henle, and distal convoluted tubule are all components of a single nephron. A collecting duct is not a part of any one nephron, but you'll notice that multiple distal convoluted tubules can connect to a single collecting duct. Okay? And that collecting duct is going to run down the renal pyramid, and down here would be our renal papilla. So all this filtrate, which is destined to become urine, is going to go through that renal papilla, which is right here, then into the minor calyx, the major calyx, the renal pelvis, the ureter, and then down to the bladder to be stored until the appropriate time for release. Okay? Let's see. I'm trying to make sure I get all of the parts on your list for you to identify. All right, so the nephron, again, is going to be the renal corpuscle, proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. Now, throughout, as the filtrates, remember the filtration happens here at the glomerulus, right? Stuff, fluid, small electrolytes, glucose, things like that. Stuff that's small enough to fit through the fenestrae and the slit pores will end up becoming part of the filtrate, okay? And that filtrate will then enter into the renal tubule. So glomerular filtration, right here, that's step 1 in urine formation. There are 2 other steps to urine formation. The second one being tubular reabsorption, and the third one being tubular secretion. Tubular reabsorption and tubular secretion is going to happen along the length of the renal tubule. And think of it this way, tubular reabsorption. We are going to reabsorb anything that got filtered out, but we don't want to lose. We want to keep. A prime example is glucose. Glucose should never be in your urine, right? Glucose is our primary energy molecule. Like, we want to have as much glucose as possible. We don't want to get rid of it, right? So, but glucose is small enough that it will get filtered out and it will become a part of the filtrate. So at some point along the length of this tubule, all of that glucose has to be reabsorbed. We don't want to get rid of it. That's tubular reabsorption. Amino acids, like small amino acids that get out, right? We don't want to lose those. We're going to reabsorb some amino acids. So it depends on, there are certain things that will always get reabsorbed, like glucose. And then a lot of the other things will depend on the environment of the body at that time, right? If we don't have a lot of something, well, then we want to hold on to it. We'll reabsorb it. Also, along the length of the tubule, you'll have tubular secretion happening. Tubular secretion is a way for our body to get rid of things faster than what filtration will allow, right? So if we relied solely on filtration, maybe we wouldn't get rid of drugs and drug metabolites or toxins as quickly as we would want to. So tubular secretion is going to be stuff, chemicals, biochemicals, or other things that didn't get out during filtration and remains in the blood in the peritubular capillaries or in the vasa recta, right? Anything that's still in the blood in these capillary beds will get secreted from these capillary beds into the tubule so that it can become part of the urine. Okay? So again, tubular secretion is a way for us to get rid of things faster than what filtration would allow. And then what happens is all of that filtrate after tubular reabsorption and tubular secretion, then that filtrate enters into the collecting duct. At this point in the collecting duct, the only thing that could really happen is we could reabsorb water. If you remember the hormone, antidiuretic hormone. Antidiuretic hormone from the posterior pituitary has an effect on these collecting ducts. So if you have low blood volume, or if you're dehydrated, you don't have enough water, well, we don't want to excrete that water in the urine. So ADH, or antidiuretic hormone, will actually cause aquaporins to be formed in the collecting ducts so that we can reabsorb water and save it, making a more concentrated urine. Okay, but again, that urine, that filtrate, which is now destined to become urine, will go through the renal papilla, into the minor calyx, to the major calyx, to the renal pelvis, to the ureter, down to the bladder to be stored. All right, so last couple of structures to identify. I encourage you to look through these pictures of these models and try to identify all those structures that I've been talking about in the videos. So next, okay, so ureters. Here, this would be our right and left ureter, which conveys urine to the bladder to be stored. Urinary bladder. So this is a sagittal section through a male pelvic cavity. This is a sagittal section through a female pelvic cavity. Here is the male bladder. Here is the female bladder. If you notice, the female bladder is much more collapsed, and that's because you'll see this big, heavy, muscular uterus that sits right on top of the, that kind of curves over and sits on top of the bladder in the female pelvic cavity. So males don't have that. Okay. So here, this is rectum, and then anterior to the rectum is our bladder. Okay. Male bladder. Down here, here's the rectum. Anterior to that is the uterus and the vaginal canal. And then anterior to that is the bladder. Okay. So let's talk about the bladder. I think I have a picture. Okay, so this is the male bladder. It's because we see prostate as well as penis, but it's a good model to show the trigon. So the trigon is the region of the bladder. It's a triangular shaped region of the bottom of the bladder, and at each corner we have an opening. Here at the base of the triangle, of the trigon, we have these two openings. This is where the ureters actually enter. So if you were to look at the posterior side of the bladder, the ureters come down and actually enter towards the bottom of the bladder. This helps create a valve-like structure, okay, so that the urine, once it goes into the bladder, it doesn't go back out into the ureters, okay? So these little holes right here, this is where the right and left ureter would connect to the bladder to bring urine to be stored. At the apex of the trigon, this opening would be where the urethra exits the bladder, okay? Let's see. Then we have the urethra. So if we go back over here to our male bladder, here is our urethra, which eventually is going to exit through the penis. Here's the urethral orifice, and that's where urine would exit, the male. Here in the female, here's our bladder. Here's the urethra. Here's the urethral orifice, which on the lateral sides of the urethral orifice, we'll see the labia minora and the labia majora. So here, this is the urethral orifice, okay? Let's see, some other structures that you need to identify. The list says Detrusor muscle. The detrusor muscle, that's a specific name given to the smooth muscle of the bladder. And the smooth muscle of the bladder is going to be oriented in, it's going to have bundles of smooth muscle all in different directions. Okay, let's see if I have any of these pictures I don't think these pictures really show it that well, but the bundles of smooth muscle of the bladder are oriented in all different directions so that you can essentially squeeze, when it contracts, it squeezes from all different directions to push the urine out through the urethra. Now, under urethra, there's a few structures that you have to identify. The prostatic urethra, membranous urethra, and the spongy urethra. Those are all parts of the male urinary tract. So here, prostatic urethra is the portion of the urethra that is surrounded by the prostate gland. Okay. Then we have this little portion of the urethra called the membranous or the intermediate urethra. It's neither surrounded by prostate nor penis. It's just intermediate. Okay. Then the portion of the urethra that is surrounded by the penis is called the spongy urethra. And this would be the urethral orifice down here. Now, when you look at male versus female, the length of the urethra of the male is much, much longer than the length of the urethra in the female. That is why urinary tract infections are more common in females. It's because external pathogens or bacteria have a much shorter distance to travel in order to infect the bladder or even go up through the ureters, eventually making your way to the kidneys. So that's why urinary tract infections are more common in females. And that's it.